the city of Wuhan, China. COVID-19 is believed to be transmitted primarily through inhalation. Once infected, most people experience mild symptoms, including sore throat, headache, fever, a dry cough, shortness of breath, and fatigue. However, in a subset of patients, the virus attacks the lungs, causing a much more serious infection. Small air sacs in the lungs add oxygen to your blood cells. This oxygen is then transported to organs and tissues throughout the body. When the coronavirus attaches to a cell, it begins to replicate within the cell. The body's immune system attempts to destroy the virus, which results in an inflammatory response that causes fluid accumulation in the lungs. As the lungs fill with fluid, the body's available oxygen decreases, which can lead to organ injury and death. The risk of serious complications increases if a person is elderly or has associated medical conditions. Once infected, a person may show no symptoms but can still infect others. What began in Wuhan has now spread across the globe.
In 1796, the scientist Edward Jenner injected material from a cowpox virus into an 8-year-old boy, with a hunch that this would provide the protection needed to save people from deadly outbreaks of the related smallpox virus. It was a success. The 8-year-old was inoculated against the disease, and this became the first ever vaccine. But why did it work? To understand how vaccines function, we need to know how the immune system defends us against contagious diseases in the first place. When foreign microbes invade us, the immune system triggers a series of responses in an attempt to identify and remove them from our bodies. The signs that this immune response is working are the coughing, sneezing, inflammation, and fever we experience, which work to trap, deter, and rid the body of threatening things like bacteria. These innate immune responses also trigger our second line of defense, called adaptive immunity. Special cells called B cells and T cells are recruited to fight microbes and also record information about them, creating a memory of what the invaders look like and how best to fight them. This know-how becomes handy if the same pathogen invades the body again. But despite this smart response, there's still a risk involved. The body takes time to learn how to respond to pathogens and to build up these defenses. And even then, if a body is too weak or young to fight back when it's invaded, it might face very serious risk if the pathogen is particularly severe. But what if we could prepare the body's immune response, readying it before someone even got ill? This is where vaccines come in. Using the same principles that the body uses to defend itself, scientists use vaccines to trigger the body's adaptive immune system without exposing humans to the full-strength disease. This has resulted in many vaccines, which each work uniquely, separated into many different types. First, we have live attenuated vaccines. These are made of the pathogen itself, but a much weaker and tamer version. Next, we have inactive vaccines, in which the pathogens have been killed. The weakening and inactivation in both types of vaccine ensures that pathogens don't develop into the full-blown disease. But just like a disease, they trigger an immune response, teaching the body to recognize an attack by making a profile of pathogens in preparation. The downside is that live attenuated vaccines can be difficult to make, and because they're live and quite powerful, people with weaker immune systems can't have them, while inactive vaccines don't create long-lasting immunity. Another type, the subunit vaccine, is only made from one part of the pathogen, called an antigen, the ingredient that actually triggers the immune response. By even further isolating specific components of antigens, like proteins or polysaccharides, these vaccines can prompt specific responses. Scientists are now building a whole new range of vaccines called DNA vaccines. For this variety, they isolate the very genes that make the specific antigens the body needs to trigger its immune response to specific pathogens. When injected into the human body, those genes instruct cells in the body to make the antigens. This causes a stronger immune response and prepares the body for any future threats. And because the vaccine only includes specific genetic material, it doesn't contain any other ingredients from the rest of the pathogen that could develop into the disease and harm the patient. If these vaccines become a success, we might be able to build more effective treatments for invasive pathogens in years to come. Just like Edward Jenner's amazing discovery spurred on modern medicine all those decades ago, continuing the development of vaccines might even allow us to treat diseases like HIV, malaria, or Ebola one day.
It's a simple gesture, one we make dozens of times for ourselves and our families to help prevent potentially life-threatening infectious diseases such as meningitis, pertussis, flu, polio. Vaccines work by preparing the immune system to better combat infection and prevent disease. They are biological products that result from a series of sophisticated and careful scientific processes that ensure the quality of each shot. Here's how vaccines are made. Production starts with growing viruses or bacteria in the lab. A wide range of parameters must be managed, like temperature, pH, oxygen rate, sterility, and homogeneity. The slightest variation in the culture media may compromise the production and cause delays. Depending on the vaccine, cell cultures take from 2 days to 3 months. From a small amount of original cell culture solution, the manufacturer can eventually produce millions of vaccine doses. The microorganisms are extracted from their environment through a process called purification, which eliminates any trace of culture media. We then inactivate the virus or bacteria. That means we eliminate its ability to cause disease. We disarm it. But we retain its ability to elicit a precautionary immune response from the body. The inactivated virus or bacteria is called a valence. A vaccine can include one or several valences to protect against one or multiple diseases in the same shot. Each valence must remain fully effective, and it must not interfere with the other valences. This careful, precise combination takes years of scientific research and development to perfect. Then, stabilizers or a preservative are added in extremely small quantities to help guarantee that the vaccine will remain stable, potent, and effective. Sometimes an adjuvant is also added to enhance the body's immune response to the vaccine. Certain vaccines are also freeze-dried into a powder to help further preserve the vaccine's efficacy. This is particularly important for vaccines that are intended to prevent outbreak-prone diseases like yellow fever, where having available stocks on hand over time is an important element to combating and containing disease. When it's time for the vaccine to be administered, it will be reconstituted by combining this powder with a diluent. The vaccine in liquid or freeze-dried form and diluents as needed are then filled into vials or syringes. When the filling phase is complete, each vial or syringe undergoes visual inspection. The quality of the contents and the container are scrutinized by both the human eye and state-of-the-art digital surveillance technology. All along the production, Sanofi Pasteur performs hundreds of laboratory tests on each and every batch of vaccine to confirm quality. In addition, samples are taken from each batch and sent to public health authorities for testing as well. Only after the vaccines have successfully passed both sets of tests can distribution begin. There are no exceptions. If a batch does not meet the quality criteria, it will be destroyed. Throughout packaging and shipping, vaccines must be stored between 2 and 8 degrees Celsius. Cold rooms and temperature-controlled shipping solutions, such as refrigerated containers and isothermal packaging, protect the vaccines during each phase of transportation and storage. A temperature tracking device is included in each shipment. Sanofi Pasteur takes care to follow strict precautions throughout the entire manufacturing process to ensure the quality of our vaccines. Production takes place in absolutely sterile conditions. Even the atmosphere is purified. Continuous control along the production chain also ensures that raw materials, equipment and manufacturing processes, and the finished product are of the highest quality. In total, more than 70% of the time required to produce a vaccine is spent on these quality controls. Depending on the vaccine, we conduct up to 1,200 different tests throughout the process. For these reasons, the total time required to produce a vaccine ranges from 6 months for the flu vaccine, for example.
So how quickly can we develop vaccines when we need them most? Vaccine development can generally be split into 3 phases. In exploratory research, scientists experiment with different approaches to find safe and replicable vaccine designs. Once these are vetted in the lab, they enter clinical testing, where vaccines are evaluated for safety, efficacy, and side effects across a variety of populations. Finally, there's manufacturing, where vaccines are produced and distributed for public use. Under regular circumstances, this process takes an average of 15 to 20 years. But during a pandemic, researchers employ numerous strategies to move through each stage as quickly as possible. Exploratory research is perhaps the most flexible. The goal of this stage is to find a safe way to introduce our immune system to the virus or bacteria. This gives our body the information it needs to create antibodies capable of fighting a real infection. There are many ways to safely trigger this immune response, but generally, the most effective designs are also the slowest to produce. Traditional attenuated vaccines create long-lasting resilience, but they rely on weakened viral strains that must be cultivated in non-human tissue over long periods of time. Inactivated vaccines take a much faster approach, directly applying heat, acid, or radiation to weaken the pathogen. Subunit vaccines that inject harmless fragments of viral proteins can also be created quickly, but these faster techniques produce less robust resilience. These are just 3 of many vaccine designs, each with their own pros and cons. No single approach is guaranteed to work, and all of them require time-consuming research. So the best way to speed things up is for many labs to work on different models simultaneously. This race-to-the-finish strategy produced the first testable Zika vaccine in 7 months, and the first testable COVID-19 vaccine in just 42 days. Being testable doesn't mean these vaccines will be successful, but models that are deemed safe and easily replicable can move into clinical testing, while other labs continue exploring alternatives. Whether a testable vaccine is produced in 4 months or 4 years, the next stage is often the longest and most unpredictable stage of development. Clinical testing consists of 3 phases, each containing multiple trials. Phase 1 trials focus on the intensity of the triggered immune response and try to establish that the vaccine is safe and effective. Phase 2 trials focus on determining the right dosage and delivery schedule across a wider population. And Phase 3 trials determine safety across the vaccine's primary use population, while also identifying rare side effects and negative reactions. Given the number of variables and the focus on long-term safety, it's incredibly difficult to speed up clinical testing. In extreme circumstances, researchers run multiple trials within one phase at the same time, but they still need to meet strict safety criteria before moving on. Occasionally, labs can expedite this process by leveraging previously approved treatments. In 2009, researchers adapted the seasonal flu vaccine to treat H1N1, producing a widely available vaccine in just 6 months. However, this technique only works when dealing with familiar pathogens that have well-established vaccine designs. After a successful Phase 3 trial, a national regulatory authority reviews the results and approves safe vaccines for manufacturing. Every vaccine has a unique blend of biological and chemical components that require a specialized pipeline to produce. To start production as soon as the vaccine is approved, manufacturing plans must be designed in parallel to research and testing. This requires constant coordination between labs and manufacturers, as well as the resources to adapt to sudden changes in vaccine design, even if that means scrapping months of work. Over time, advances in exploratory research and manufacturing should make this process faster. Preliminary studies suggest that future researchers may be able to swap genetic material from different viruses into the same vaccine design. These DNA and mRNA-based vaccines could dramatically expedite all 3 stages of vaccine production. But until such breakthroughs arrive, our best strategy is for labs around the world to cooperate and work in parallel on different approaches. By sharing knowledge and resources, scientists can divide and conquer.
It's a rather simple plant. We can make things like polio vaccine. Really quite extraordinary. Plants are fascinating organisms because they almost grow from nothing. I first started proposing plant-based methods for making vaccines around 1990, and actually people laughed. It's wonderful to see that people don't laugh about it now. The amazing thing about this approach is it combines science with what is essentially gardening. And we've come to this stage where it actually might save human lives.
So is this part of your usual science getup?
Yeah, this is why when I'm actually showing people things, so people can instantly recognise me as the head honcho. I wear a tiara. I'm George Lomonosov. I'm a project leader in the Department of Biological Chemistry at the John Innes Centre in Norwich. The John Innes Centre specialises in plant and microbial science. I actually joined it about 37 years ago, and so I sometimes get the feeling I'm part of the furniture. So vaccines fool the immune system to protect you against disease. And what you do with vaccination is you take something which looks like a disease-causing organism but actually isn't. We can call it a surrogate or mimic, and that fools the immune system into making antibodies, and those antibodies will actually protect against the real infectious organism. The problem is that it takes traditionally many years to produce a new vaccine against an emerging disease, and we don't know what the next one to emerge will be. So the great thing about plant-based production is you can identify the virus and extremely quickly, in 3 to 4 weeks, go to making new vaccines. We use the plants to make just the parts of the disease-causing organism which can stimulate immunity, but don't have any of the weapons which actually cause the disease itself. You can see I'm dressed in a rather stylish way, ready for a dinner party, wearing this rather fetching hairnet. The plant we use is Australian dwarf tobacco. This is an Australian dwarf tobacco plant. They grow like teenagers from now on. In a week's time it'll be right. Another week it'll be too big. So you have to get them just right. A bit like Goldilocks. We use this plant because it has a defective plant immune system, which means it's easy to produce new vaccines in it. The technique we use is called transient expression, where you infiltrate the leaves with the material for the new vaccine and you'll get production within about a week. We take the leaves and then blend them and then use that material to isolate the new vaccine. It's an incredible thought, really, that something so simple as this and so cheap to grow can actually be a lifesaver. A lot of emerging diseases appear in less developed parts of the world.
The Ebola outbreak in West Africa is spiraling out of control.
This is the deadliest outbreak of Ebola on record.
At the time there was no vaccine, but fortunately some colleagues in the United States were exploring the use of plants to make a therapy for the treatment of Ebola victims. The development of ZMapp offered a glimmer of hope and showed how such therapies could be developed in the future.
A handful of people were successfully treated with ZMapp.
Today is a miraculous day. I'm thrilled to be alive, to be well, and to be reunited with my family.
It's fantastic to see that fundamental science being used to increase the health of the population throughout the world. This technology is faster, safer, more readily scalable, and can be used reactively to combat emerging diseases. This approach that we've developed is particularly suited for production in the countries where the vaccines are most needed. And it's absolutely amazing to see how that work can help save lives.
The World Health Organization has listed 4 plant-based vaccine candidates to tackle the coronavirus. The list includes the vaccine candidate that is being developed by Canadian firm Medicago. This company is using plant-based bioengineering methods to develop a vaccine to fight COVID-19. At the moment, the vaccine is in its second phase of clinical trials. How does it work? Plant-based biotechnology harnesses the speed, flexibility, and efficiency of plants to come up with vaccine candidates. Rather than replicating a virus, it aims to engineer a virus-like protein in living plants. A virus-like protein, or VLP, is a non-infectious version of a given virus. Medicago used a type of tobacco plant for this purpose. When administered, a VLP mimics a virus and is recognized by the immune system, thus eliciting a protective response. Since it lacks a genome, it is seen as completely harmless during human trials. Medicago claimed a 100% success rate in phase 1 of its clinical evaluation. Can a VLP be used to produce a quick vaccine for human use? The answer is no. It would not be possible to do so, especially in pandemic situations where there is a demand for a swift solution. For this purpose, Medicago's candidate is added with adjuvants from GlaxoSmithKline and Dynavax. What is an adjuvant? An adjuvant enhances the immune response to viruses. This helps to ensure stronger and longer-lasting immunity against infections that can be achieved by the vaccine alone. What are the advantages of such initiatives? Using plants as biofactories has several advantages. They can be produced cheaply in very high amounts. Carrier plants such as potatoes and corn are readily accepted by patients. Also, the antigens derived can be stored for longer periods of time.
In the fight against coronavirus, researchers all over the world are racing to develop a vaccine for COVID-19. Developing one quickly and in large quantities remains a huge challenge, but it's hoped molecular farming, turning tobacco plants into so-called biofactories, could speed up the production of tailor-made vaccines and antibodies. Here at the Institute for Plant, Molecular and Cellular Biology in Valencia, top scientists are attempting to harness the power of nature. Using the latest techniques to cultivate and sequence the genome of the Nicotiana benthamiana plant, researchers are able to produce tailor-made molecules to fight disease.
Muchos de los medicamentos que usamos hoy en día están extraídos de plantas.
Many medicines we use today are extracted from plants, but now thanks to biotechnology, we're able to manufacture other substances from plants different from those which they produce naturally. We do that by introducing genetic material into their genome. This contains information that enables the production of medicines such as antibodies, vaccines, and other products.
Vacunas, anticuerpos y otros productos.
The plant is native to Australia and has been used to produce vaccines and antibodies for viruses such as Ebola. By injecting other DNA into its leaves, its genes can be modified to create specific pharmaceutical products in large amounts.
We give the plants instructions to improve them, to change specific genes. For example, we are interested in having plants that do not flower. We want to grow plants that produce more biomass because those are the kinds of things we want the plant to do.
Hopes are high that by harnessing the plant's cells and sap, the technique could eventually help pave the way to producing a COVID-19 vaccine. Experts say the approach has several advantages. It offers the means to produce large amounts of the required viral protein, is low cost as well as safe.
Plants are very useful for researchers in that they are another means of expressing viral proteins or nucleic acids without having to actually work with the causative virus itself. And we can use techniques of synthetic biology to make things like virus-like particles without handling the infectious virus.
The modified plant can generate proteins similar to those found in a human cell. Researchers say a growing number of companies are embracing this technique.
Ci sono circa in questo momento un centinaio di gruppi.
There are about 100 groups in the world that are trying to develop SARS-CoV-2 vaccines.
Ci sono anche alcuni.
Some of these groups and private companies are using this plant, whose genome we have sequenced. Making the genome available will help these firms optimize this production.
Da parte nostra del genoma prima della pubblicazione presumibilmente servirà a queste ditte per ottimizzare questa produzione.
By making the genome publicly available, scientists at the EU-funded project hope this will speed up the search for new products in the fight against this and future pandemics.